So, time to begin the transcription translation process. All right, so what do we got? Well, first of all, let's take a look at where everything happens in the model. And hopefully you've watched the animation so you get a feel how this works. But basically, you've got your DNA in the nucleus. The DNA is going to unzip just like it did for replication. The helicase molecule does the unzipping. And this time, instead of DNA polymerase reading the code to bring in the appropriate nucleotides and then bonding the nucleotides together, it's the polymerase called RNA polymerase. So it's the enzyme that reads the code, brings in the appropriate nucleotide, and bonds it all together to form this messenger RNA strand. Now you can see messenger RNA is a single strand compared to DNA, which is the double-stranded helix. And because it's a single strand, it's actually small enough that it can pass across the cell membrane out into the cytoplasm, and it travels to a hookup site on the ribosome. Okay, so it specifically hooks up on what we call the active site on the ribosome. And it's going to read it through a lot like if you take and feed fabric through a sewing machine, or you see a ticker tape where it spits it out and after it's reading through the machine. That's exactly what happens here. The code would enter on this end, and it would be kind of scooted along, and the code would be read. Now remember, it reads in 3 nucleotide codon sequence. So you can see there's 3 nucleotides right here. Each one of these little bumps sticking up is representative of a nucleotide, and that would be corresponding to a letter like A, T, C, or G. What's going to happen is the transfer RNA molecule, which is this whole piece right here, is then going to bring in the appropriate base pair that matches up. So if it's an A, it's going to be a U. If it's a T, it would be an A. If it's a C, it would be a G, and so forth. Matches it up. And then attached to the transfer RNA molecule is a protein, is an amino acid. And that amino acid, if the code is correct, would then be matched up and bonded to form the protein chain. Sometimes we call that the polypeptide, right? And then the transfer RNA molecule would move off, and another one would come in and read the next 3 letters. And that would just keep happening until the entire protein chain would be assembled. Okay, so that's basically how the process works. 3 types of RNAs then that you definitely need to know by name, by structure, and by function. Okay, messenger RNA, we know it as mRNA, stands for messenger. It's the one that's couriering the code. Okay, it's going to be single-stranded, and it's going to carry the DNA code out of the nucleus, because it's a single strand, into the cytoplasm. It can pass across that membrane. Our RNA, which is known as ribosomal RNA, is found in the ribosomes. Makes sense, right? And it's responsible for, well, first of all, the ribosome itself is composed mostly of this rRNA, and it's going to be responsible for when that amino acid is released from the transfer RNA molecule to make the polypeptide bond that holds all those amino acids together in that protein chain. The third RNA is going to be transfer RNA, or tRNA, and it is going to be the molecule that brings in 3 base pair nucleotides that match with the messenger RNA strand, and we're going to get into the codon, anticodon thing here in a minute, to read the code, to transfer the code, and then release the amino acid that it's carrying on it so that it can then be bonded by the rRNA. So that's basically what's happening. You need to know all 3 of those RNAs and what they're responsible for. So this table will be crucial. These are, remember we did the script for replication today. Well, this is the kind of the transcript for the steps of transcription. So what's going to happen? Messenger RNA is going to carry the code that will be translated to form the protein. Okay, and remember, it's read in 3 nucleotide chunks, if you will, 3 nucleotides at a time, and that's called the codon. Ribosomal RNA is going to make sure that the bond forms between each one of those amino acids, okay, and it's located on the ribosome. And transfer RNA is going to have the anticodons, which is the 3 nucleotides that would base pair with the codon. That's why it's called the anticodon. Okay, all important to know about the transcription process. That's not the steps. We're going to get into that in a minute. All right, I walked you through kind of the steps with the other model, so I'm just going to highlight some of the key things here. You've got the DNA polymerase. Well, first of all, helicase is going to unzip. DNA polymerase is going to bring in the nucleotides and proofread to make sure that the code is correct. That's kind of the first step. Okay. Then messenger RNA is going to leave the nucleus and head to the ribosome. Remember, I talked about the codon. So on the messenger RNA, you've got 3 nucleotides that are chunked together, okay, and that's called a codon. And in this case, the messenger RNA code is AUG. And I'm going to show you how to use the amino acid wheel to find out what amino acid would be brought in by transfer RNA for this AUG code. Okay, it happens to be methionine, or we use the abbreviation MET. Then it would read, and that actually is a start codon. So that's the one that tells the protein is going to start synthesizing. The next codon would be CUU. Transfer RNA is going to bring in the GAA to base pair with it. And because the code is CUU, if you look that up in the amino acid wheel, you would see it would bring in the amino acid leucine, okay, or the abbreviation LEU. So that's going to be the actual first amino acid in our protein synthesis, or in our protein sequence that we're going to make the polypeptide. All right. This is where I said, remember, it hooks up at the ribosome at a very specific site. Well, this is that site where it's happening, okay? So here's your entire ribosome molecule. Here's the binding sites where those 3 codon nucleotides are going to be read. There's a lower portion and an upper portion. You don't really need to know all the details of that, but just know that it kind of like gets sandwiched right between there like a sewing machine, and so it reads just like the fabric being fed through a sewing machine. This is what the anticodon would look like. You'd have this transfer RNA molecule. Okay, it's going to have the 3 base pair nucleotides that are going to match with the codon. Okay, this is called the anticodon, and it would base pair with the codon that would be down here on the messenger RNA strand, and then it's going to release the appropriate amino acid. This animation kind of shows the entire translation process as it goes through. You can see the strand has left the nucleus and it's traveling through the cytoplasm. It's being fed through multiple ribosomes at the same time, making multiple protein chains at the same time. And that's very likely what's going to happen, is you'll have lots of work going on that strand at one given time, making lots of different proteins. I mean, we need lots of proteins in our body. So the appropriate transfer RNA brings in the nucleotides to base pair with the messenger RNA strand. It then corresponds to the appropriate amino acid, and it's released, and the protein chain, or the polypeptide, is then made as you see here. They can be pretty long strands, or they can be pretty short. Important to remember that the bond that forms between amino acids is the peptide bond. This animation is basically just showing you everything else the same though. This is your messenger RNA strand. Here's your 3-letter sequence. That's your first codon. Okay, it's AUG. Remember, it just happens to be the start codon. So if you look this up in the amino acid wheel, it would be referenced to methionine. And UAC are just the base pairs that are perfectly going to match with, according to our base pair rule, the AUG. All right. This is how you figure out what amino acid is going to be paired with which code. Like, for instance, we were looking at AUG, which was our start codon. So in this configuration, this is a table, and this one's nice because it gives you the whole letter sequence. But there's a wheel also that you can use, and you can pick whichever one you want to use. The textbook has the wheel in it, just FYI. So the first letter was A, the second base was a U, and then the third letter was a G. That's not right. A, U, and then G comes over here, and it meets up where they all intersect is methionine, right there, MET. So that's how you know that's the amino acid that would be coded then for that base codon sequence. And that's the amino acid that would be released. This is the wheel I was talking about, the same effect. You start with your inner wheel and then go to your second layer and then your third layer. So it would be A, U, G. So I think this one's easier to use, but a lot of students actually prefer the other. All right, there's just another example to show you how the transfer RNA molecule is bringing it in and how the protein synthesis occurs. Process I've already described for you. Some interesting facts then to kind of give you an idea. Somebody asked me the other day, and I don't know whether it was first block or second block, but how fast does all this happen? Well, here's a statistic for you. The protein chain, which is going to make keratin, which is what your hair fiber is, basically it's dead cells, and that all that's left is this protein fiber keratin that gets fused together to make the long strand. But the cell, the hair follicle cell that's located at your scalp, in order to make a new cell, the keratin protein chain that it would make is 400 amino acids long, and it's assembled in 20 seconds. So you can see that that entire process happens very, very quickly. So just some interesting tidbit information on this slide for you for the future. So that is your translation transcription notes for this session. I'll see you in class tomorrow for the quiz.